for nanomaterials for sensing and remediation. Unlike the last excellent speaker, I will be concentrating on the applications rather than the theory, but we can discuss the theory and questions if you like. So my team has been working on nanomaterials and nano embedded coatings for chemical sensing, catalyst and biomedical applications. All our materials are based upon quantum dots or carbon dots. The principle is very simple. Broadband radiation impinges on a dot. The band gap in that dot then electrons are activated across the band gap, and very sharp radiation that can be used for identification then comes out of the dot. We can put these dots in, use them separately, or we can embed them in membranes. And obviously we look at how the local environment changes their properties, and more recently, how they can react with active species to be catalysts. So the applications that we're talking about is contamination in water, monitoring and antimicrobial activity in biosystems, and a catalytic remediation of pollutants. Now we're going to do a double header in this presentation. I'll give an overview of our team's applications, and then Sabrina, as Felipe mentioned, one of our senior PhD students, will go into one particular application. Okay, so firstly, our work started predominantly on water sensing, particularly on sensing heavy metals in water, because these can be a significant toxin, particularly for women in pregnancy and young children. And so particularly we want to determine the level of lead, mercury, chromium, and other heavy metals. We started with semiconductors, cadmium, cerium, zinc sulfide, and we got quite a long way with those. They have a very high fluorescence yield, generally between about 60 and 80%, and so they are very efficient. Unfortunately, they are also mildly toxic, and there's a perception, a greater perception, the reality perhaps, of their toxicity. So we have since moved on to using carbon dots. These carbon dots we can make from a whole lot of natural sources: bread, prickly pear, lemon juice. In fact, Sabrina will talk about some work from prickly pear. So they are very natural, they have no toxicity, and they are extremely cheap. The yields are much lower though. The yields are somewhere between 5 and 25%, depending on the dot. The other issue with the carbon dots, it's very clear what promotes the fluorescence. With the semiconductor dots, it's much more variable about the carbon dots. It's a combination of the band gap and the surface molecules. So the basic simple principle is we take whatever type of dot, we functionalize that with various ligands. We can choose the ligands so they are sensitive to particular analytes, in our case, particular heavy metals. And then the fluorescence then drops and in the presence of that heavy metal. And we're showing some work on lead in the far right hand side, and you can see as we increase from 0 up to 0.06 approximately micromolar, etc., we drop the fluorescence, and we can use that to detect the lead. Over on the left hand side, we're showing the sensitivity of that system to a range of different heavy metals. And so the system we're showing here is actually one which is very sensitive to copper, but in that it's also slightly sensitive to other metal ions. So we can get a great deal of sensitivity out of these nanodot sensing, but we do have a problem with selectivity. It's harder to distinguish between the different metal ions. Another system we have developed is putting those dots in gels and then using those gels to detect chromium. This on the right hand side here was just showing the viability of cells. So there's very little effect of our gels or our dots on cells life. Then we're doing a fluorescence measurements in a multi-well device, coming up with our sensitivity of this particular system to chrome, and again then showing the relative sensitivity, highly sensitive to chrome, chromium ions, but slightly sensitive to other elements. Sometimes this won't matter if we suspect that we do have a high chromium 6 level, but if we want a general sensor, this slight sensitivity can be a bit of a problem if there are multiple ions of widely different concentration. So what do we do in order to get greater selectivity? One way we can do that is use a different range of quantum of either quantum or carbon dots. In this case, we're using 6 different functionalized carbon dots. They all have slightly different responses to the heavy metals, and then we can do some factor analysis to look at how those groups of carbon dots, in this case everything but C3 and C5, and in this case C3 and 5 respond, and we can then cluster the responses, and then we can get good separation between the heavy metals and use this analysis built into the programming to be able to get much better selectivity. So that's one way to approach the problem. The other way to approach the problem is by molecular design. This is not dots, but at just fluorescent molecules based around the Cornwall family, and we have derived a large range of slightly different molecules. What we're doing here is we're altering the attachment to the metal ion. And in this case, we're looking at how these different isomers attach to copper 2 ion. And they generally will bind into this point in the middle of, say, 2 in that mouth there. And the degree of binding of the heavy ion is controlled by the proximity of ions that can share lone pairs, by the amount of flexibility in the molecule, by the size of the gap they can bind into, and by any steric hindrance. So we're playing with all these factors in this series. So by looking at a range of molecules, playing with those binding factors, we can then select out the 2 molecule forms, which are these 2 forms, 2 and 4, which have the highest sensitivity and the highest selectivity. We can then redesign these binding sites to give selectivity and sensitivity to other heavy metals. So that is, I guess, the nano side of it. The engineering side of it is normally, if you want to sense water, you have to take it to a lab, to IC or ICP-EMS, and that is very expensive. It takes quite a lot of time. So to get away from that, we've designed a small portable device, which is an inbuilt fluorescence. The device itself is based on simple electronics. It cost about $150 Australian to make, so even less than US dollars, and it costs about $1 to do a test. So we simply put an ethicate of solution in here, and then we have the light coming across and our dots in the ethicot, and we can get sensitive in situ measurement of heavy metals. So that's the sensing side. We're not just interested in environmental sensing, we're also interested in biological sensing. And this is a case where we've produced carbon dots, in this case from bread, and we have cultured muscle myoblast and myotubes in the lab, and then put the dots into that culture and observe that the dots can cross the membrane, and then they can illuminate the features of the myodots and the myoblasts. We haven't yet done this in animal trials, but we have shown that we can cross cell boundaries and we can illuminate biological structures. We can do more than simply illuminate, though. This work is looking at, can we actively reduce bacterial infections? And so we have not dots this case, but magnesium hydroxide nano sheets. This is just an image, macro image of those sheets. This is showing their fluorescence, and this is putting those sheets in a bandage, which we've made by integrating the fibers. What then happens is that at particular pHs indicative of inflammation, the magnesium tubes give off magnesium ion, which significantly reduces the fluorescence. However, in addition to reducing the fluorescent, reducing the microbial activity, and therefore reducing the inflammation in the wound. So in addition to reducing the inflammation in the wound, they also fluoresce, which is what we're showing here, the fluorescence of the particles. And we can use that fluorescence to tell us that the active ingredient, these nanosheets in the bandage, are still active. This is just some data showing how the magnesium release from the fibers affects two different types of bacterial agents, MRSA and C. albicans, showing that we get a lot of bacterial death, in fact, almost total bacterial death, by the release of the magnesium hydroxide. And this in the opposite is showing that it has very little effect on human cells or human cell analogues. So we can go past simply sensing with the nanostructures. We can both sense and then have intervention against microbial activity. We can put this actively into bandages and other structures to assist medical application. At this point, I'm going to pass over to Sabrina to go into detail on one particular application, which is environmental remediation.
Okay, thanks, Ivan. Hope you can hear me. Is that all right?
Yes, we can hear you, Sabrina.
Yeah, good afternoon, everyone. Good. Good afternoon. I'm Sabrina. I'm a final year PhD student at our RMIT University, and I'm going to present to you an overview of my PhD project, which is on the catalytic remediation of environmental pollutants by carbon dots. Next, please. As mentioned by Professor Ivan, due to the carbon dots is rich in functional groups, the surface charge and the increased surface area, they are potential candidates as nanocatalysts in the Fenton catalysis of a range of pollutants, environmental pollutants. So the Fenton reactions are actually part of the advanced oxidation processes, the AOPs, which are reactions at the bottom, catalyzed by ion 2 and hydrogen peroxide in order to generate hydroxyls, which are highly reactive and oxidizing agents that can ultimately and consequently lead to the destruction of the organic pollutant. However, there are some disadvantages in these such as the need of an acidic pH and the toxicity of the sludge formed during this reaction, etc. To overcome these drawbacks, the Fenton-like reactions are a promising and alternative traditional Fenton reactions. So as you can see in the reaction equation on the bottom, the ion 2 can be replaced by ion 3 or metal ocean metal. So these Fenton-like catalysts have been successfully used in many investigations for environmental remediation. Next, please. So nevertheless, there are still some important risk gaps that haven't been addressed so far in the field, such as the catalytic properties of dots in the presence of peroxide are unexplored for the environmental remediation. Also, the efficacy of the dots made of green material, as mentioned by Professor Ivan, these dots can be made by plant extracts, food waste, and other of natural resources. So the efficacy of these kind of green synthesized dots that are able to act as Fenton-like catalysts in degradation of environmental pollutants, such as commercial dyes, is unknown. And also another very important aspect is the ecotoxicological aspects of the effluents generated during the catalytic remediation containing the dots, especially using Fenton-like reactions, are also unexplored and are important as well. So there's some limited information about this. So therefore, the main aim of my PhD research is to investigate the use of the nanocatalysts in the Fenton-like reactions in the degradation of dyes in water. Next. So as our first research experiment, we aimed at answering some questions. So can green synthesized act as Fenton-like catalysts in the decolourization of dye? And what are the key catalytic properties of the dots? So briefly, we synthesized the dots from prickly pear extract as our first step using a hydrothermal approach. And they were further characterized using a set of characterization techniques such as FTIR, XPS, TEM, DLS, and photoluminescence and UV-Vis studies as well. And finally, they were tested as Fenton-like catalysts for the decolourization of 3 different dyes, so crystal violet, methyl green, and basic fuchsine. So this study was published last year. Next, So in terms of characterization, the XPS and the FTIR showed incorporation of surface functional groups from the precursor into the surface of the prickly pear dots. So surface functional groups such as carboxylic acids, hydroxyls, and amines. And such multifunctional groups could make a potential candidate, make the carbon dots a potential candidate for the catalysis in Fenton-like catalysis, as suggested by the literature. So the TEM here shows near spherical dots, and the DLS confirmed the size of the dots, which were around 13 to 20 nanometers. And in the figure 3A, we have the zeta potential measurement. So the surface charge of these dots was assessed using zeta potential measurements, and the analysis revealed an increasing negative surface charge with an increasing pH. And next, the catalytic characterization of dyes by dots was tested under different conditions, such as different carbon dots concentrations, different pHs, different temperature, and He2O2 concentrations. So this reaction was actually optimized nearly 90% of the mix of the dyes. So they were actually tested as individual dyes and also as a mix, like blended, and nearly 90% of the mix of the dyes was removed from the water solution under mild conditions using a pH near neutral, 20 degrees and 0.25 milligram per mil of these dots and 0.5% of H2O2 in only 60 minutes. We went there studies, similar studies using Fenton-like nanomaterials. Yeah, this work presents some advances over these studies. Next, please. Also, to investigate the mechanism of action,
Hi.
yeah. So in order to, yes, oh good, investigate their mechanism of action, some scavengers such as ascorbic acid, methanol, as you can see in the figure 6a. So these scavengers, they were added into the reaction, so they would quench any production of reactive oxygen species being formed during the catalytic remediation, and then would consequently decrease the decolorization of these dyes, which actually happened. So these analyses showed that ROS such as singlet oxygens, hydroxyls, and superoxides were being formed during the reaction, which gives us an insight of the mechanism of action. Moreover, the zeta potential in figure 6b revealed that the reduction of the surface charge after the exposure. So we measured the zeta potential before and after H2O2 to see if the charge was changed. And in this near between 6 and 8 pH value, there was a significantly decreased zeta potential measurement, which just reinforced the role of the surface charge during this decolorization reaction. In addition, not shown here, the XPS and FTIR also showed reduction of peaks for carbon double oxygen, carbon single nitrogen, carbon single oxygen groups and bonds after the exposure to H2O2. So this also helps us to understand the mechanism of action involved in this catalytic remediation of the dyes. So as conclusions for these works, the dots were successfully synthesized using a very easy hydrothermal approach, using a prickly pear plant as a carbon precursor. And also the reactive oxygen species, such as the singlet oxygens, superoxides, and hydroxyls were formed during the reaction. They played a major role in the catalytic behavior of the system. And these nitrogen and oxygen groups, they participated, and they have a major role as well in the catalytic decolorization of the dyes. Next. Please. So in our second research experiment, we aimed at investigating the functionalization of the dots and their effects on the degradation of the dyes. And also we aimed at assessing the ecotoxicological impacts of the effluents generated during the catalytic process. So briefly, for this experiment, we used the ferric citrate, urea, and citric acid as precursors because we wanted to analyze the doping of the dots using ion 3. So the same procedure was performed using a hydrothermal approach to make these dots, and the same set of characterization techniques was used. And for this one, used methylene blue as a model pollutant, methylene blue dye as a model pollutant. And finally, we performed an ecotoxicity test using a macrotoxicase standard ecotoxicity test for water. So this is a study with catalytic degradation of methylene blue using ion nitrogen carbon dots, and it has been submitted to a journal and is currently under. Next. So the characterization of the ion and containing dots revealed the presence of many surface functional groups. As you can see in the FTIR, when we compare the precursors to the carbon dots containing different ion contents. And the literature reports nitrogen, oxygen, and ion groups, which were the ones found in the FTIR spectrum. They can introduce different functionalities, and they can also create more active sites over the carbocatalysts. Well, and in terms of charge, the dots were mainly negative across of pH values, as you can see in the figure in the graph set 7b. In turn, they were mainly negative, and the most negative zeta potential was observed on at the pH 8. Also at the bottom, you can see the TEM that showed they were near spherical in morphology and an average size of 7 nanometers. Yeah. Next. Yeah, so the methylene blue dye degradation studies showed a positive relation between the activity and the ion of the dots. So the Fenton-like degraded dots containing the highest content of ion resulted in almost 100% removal of the methylene blue dye in only 6 minutes, as you can see in the graph 9, under a pH 8 and at 50 degrees. Moreover, not shown here, but we also did some FTIR and XPS analysis when the dots before and after exposure to H2O2. And this analysis showed that those dots exposed to H2O2, they revealed a reduction on peaks of nitrogen and oxygen groups, which could also help us to explain the mechanism of action and what's similar to our further experiments with the ones. And finally, the ecotoxicity results were expressed as EC50. So EC50 means a statistic estimate of the concentration of a chemical, of the toxicant, that is necessary to produce an effect in 50% of a population. So in case we used marine bacteria called the Levibrio fisheri. So these analyses revealed that the catalytic degradation performed by the dots to using only hydrogen peroxide. So the dots did not generate harmful byproducts to aquatic organisms at 24 hours, and therefore may represent an alternative treatment for wastewaters that can significantly reduce the toxicity of the effluents. Next, please. So as conclusions, carbon dots were successfully synthesized via an easy, economical, and green route, which generated 7 nanometers negatively charged, not reaching amines, ions, and carboxyl functional groups. The spectroscopic analysis revealed that nitrogen, oxygen, and ion groups participate as potential active sites during the reactions, and the use of the technology, the dots, resulted in a significant reduction in the toxicity of the effluent. Next, please. And conclusions of today's presentation. So for bio and environmental sensing and intervention, carbon or non-semiconductor structures are preferred for toxicology reasons and have sufficient performance. Oh, sorry, can you please just back to the, yes, thanks. And for sensing, low detection limits can be achieved, but issues remain with specificity, but molecular design cross-analysis can assist with that. The dots can migrate and illuminate biological organisms. The nanostructures can play multiple roles, both antimicrobial and sensory roles. And the carbon dots can be designed by functional and then reduce the toxicity of the effluents of environmental pollutants. And some perspectives of future studies in the field in our group and potential collaborations are the possibilities of multifunctional dots that can combine sensory and active functions are significant, and this technology could be applied in a variety of other hazardous contaminants in a large scale, cost-effective, efficient, and feasible and green remediation approach. And the dots, this technology, they could be embedded in other types of materials such as nanofibers, membranes, and etc. for water treatment.
Thanks, Sabrina, and Felipe, over to you to any questions.